We're going to be looking at Life Sciences Paper 2 from November 2022. So let's start with Section A, Question 1. Always, you're going to have your paper starting with a set of multiple choice questions. And I don't want you rushing through your multiple choice questions. There's a very certain strategy that you need to use. You need to read the question, you need to think of what the answer is, and then look at the options so that you're not misled by the options. Go with your gut feel, first of all. So let's begin. 1.1. Various options are provided as possible answers to the following questions. Choose the answer and write only the letter next to the question. So you would write the question number and then you would write the letter nice and clearly, block capital letters. So our first question, who was the scientist who discovered the fossil Karabo? Or Karabo, remember, is the common name or the familiar name of Australopithecus sediba. So who discovered this fossil? Was it A, Robert Brown, B, Lee Berger, C, Raymond Dart, and D, Ronald Clark? Well, Dart, in fact, Brown and Clark were scientists working quite a few decades ago in the field of paleoanthropology. You would remember that Karabo, or Australopithecus sediba, was discovered fairly recently, and Lee Berger is the scientist. In fact, it was Lee Berger's son who was the first one to discover the fossil, called his father's attention to it. So our answer for 1.1.1 is going to be Lee Berger, B. Which one of the following is a source of variation? When we mean source of variation, something that is going to produce variation that occurs during normal meiosis. Read your question carefully. We're looking at meiosis, not mitosis. And the options are A, random mating, B, random arrangement of chromosomes, C, chromosomal mutations, and D, cloning. Now, we go through that list, eliminating the obviously incorrect ones. The obviously incorrect ones are going to be random mating, for example. We're asked specifically during meiosis, and mating happens way after meiosis, so that is an incorrect suggestion. Another completely incorrect suggestion is cloning, because cloning is something that we do. We are not concerned with meiosis at all. We take already formed cells, not gametes, and we work with those cells in terms of cloning. So let's look at B and C. Random arrangement of chromosomes. What we're referring to there is metaphase, and in particular metaphase 1, because we're referring to whole chromosomes. And remember that on our imaginary line going through the cell, the center of the cell, we're going to see in metaphase 1, we're going to see homologous chromosomes lined up along the equator. And in some instances, these chromosomes are, for example, let's get another color here. This one could be the maternal chromosome. In other words, the chromosome from the female or the mother individual. And sometimes we will see both maternal chromosomes on the plane that is going to move to a single pole. Or we could see that in some cases the maternal chromosome is going to go to the top pole, and the maternal chromosome, this one, is going to go to the bottom pole and not the top pole. So it's a complete randomization in terms of the arrangement of homologous chromosomes on the equator. That most certainly is going to bring about variation, and it is also part of meiosis. So option B satisfies both of the requirements. Chromosomal mutations refer to chromosomes probably moving to the incorrect poles or parts of the chromosomes breaking off, and those things don't necessarily happen during normal meiosis. We must remember that chromosomal mutations are abnormal. They are when meiosis or any other process happens abnormally. And so our answer most definitely is B. So on your answer book, 1.1.2, and a nice clear B. How many sex chromosomes does a normal female human inherit from her mother? So we need to remember what we term sex chromosomes. Our sex chromosomes are the X and the Y chromosomes. In a female, we will have 2 copies of an X chromosome, one received from the mother and the other one received from the father. In a male person, we're going to have XY. The Y is going to be received from the father and the X from the mother. So the mother is always going to donate an X chromosome. In the case of the father, it could be the X chromosome or it could be the Y chromosome. However, our question asked how many. What number of sex chromosomes does the female inherit from her mother? And there we are. She will only inherit 1 sex chromosome from her mother. So our answer to 1.1.3 is going to be A. During which phase of meiosis does the nuclear membrane disappear? So we start off with a cell that has a nuclear membrane around the contents of the nucleus. So it'll be chromatin network, it will be nucleolus, all inside this membrane, which is the nuclear membrane. But at the start of meiosis, we're going to see that nuclear membrane breaking down, and we're going to see our chromosomes starting to separate out as individual chromosomes rather than the network. And we're also going to see that the nucleolus disappears. So our question is saying, during which phase does this nuclear membrane disappear? And we're offered 4 phases: metaphase, telophase, prophase, and anaphase. Now, I told you that it's at the start of meiosis, before we in fact start dividing the cell, that nuclear membrane has to disappear. So what you need to do is you need to put these phases in order, and it's always prophase followed by metaphase followed by anaphase followed by telophase. I want you to think of this really naughty cat. Okay, the cat peed on the mat. Will you remember that? And that helps you to get your PMAT, your prophase, metaphase, anaphase, and telophase in order. Now, during which of those phases is the nuclear membrane going to disappear? It's going to happen at the start of meiosis, which is prophase. So on our answer sheet, we're going to write 1.1.4, and prophase is option C. 1.1.5. Which one of the following is an example of discontinuous variation in humans? Now, you've learnt about two kinds of variation: discontinuous and continuous. So continuous variation is when you have a range of options. If I wanted to come to your class and work out the height of the different people in your class, I would be able to arrange them from shortest to tallest, but there would, in between the shortest and the tallest, there would be a range of heights. That's something that we call continuous variation. So someone might be 1 meter 52. Somebody else is going to be 1 meter 52 and a half, or 1 meter 53. So there's a continuous range of phenotypes. But in some of our phenotypes, it's a this or a that. It's discontinuous. So you're either blood group A, blood group B, blood group AB, or blood group O. You can't have an alternate one that is somewhere in between blood group A and blood group AB. There's no A and a half, for example. So you have to look at different examples of phenotypes, and you've got to work out, is there a range of phenotypes, or is there this or that? That's all. And if it's a this or a that, it's discontinuous. So let's look at the options that we're given. Height, which I've already explained to you, is continuous variation. What about heart rate? Heart rate, we could measure everybody in your class, and we could say some people have slower heart rates, some have faster heart rates, and some have their heart rates somewhere in between. So once again, this is an example of continuous variation. And the same comes down with weight. There could be something that weighs or has the mass of 1 kilogram, and then something that has a mass of 10 kilograms, but there are options in between, and so that is continuous. When we talk about gender, we're saying that there's either going to be a double X or there's going to be an XY. So it's either going to be a female characteristic brought about by 2 X chromosomes, or it's going to be a male gender brought about by XY. Of course, we know that there are mutations that can occur where we have XXY, for example, but we're not looking at mutations. We're looking at examples of discontinuous variation in humans in general. It doesn't say anything about mutations. And so our answer to 1.1.5 is C, gender. For a particular characteristic, the offspring inherits. What do we inherit? 1 allele from the mother and 1 allele from the father. So let's have a look at what we've got here. We've got 2 homologous chromosomes that form a pair. Let's imagine that we're looking at chromosome 10. This one you inherited from your dad, and this one you've inherited from your mother. And along the length of the chromosome, we are going to find different genes. And each gene is going to code for the same characteristic, but there could be variations of it. So at locus number 100, we could find, for example, the gene that codes for blood type, and it could be that the allele that the mother has given you is for A. So it would be immunoglobulin A. That could be the allele from the mother. The allele from the father is also going to code for blood type, but that allele could be different, or it could be the same. So it could be IB. It could also have been IA. It could also be little I. These are variations of a particular gene, and variations of a gene we call alleles. So getting back to our question, does the offspring inherit one of those alleles from the mother and one of those alleles from the father? Absolutely. So it looks like the answer could be option A, but we don't leave it there. We read through the other options just to be sure. Does the offspring inherit both alleles from the father or both alleles from the mother? That would be a problem, because it would be inheriting then 2 alleles from the father or 2 alleles from the mother, and we know that our homologous chromosomes separate during meiosis. So both of those options are impossible. And what about option D? The alleles are inherited from either the mother or the father randomly. And that is also problematic, because there is no inheritance randomly. So it means that this allele is inherited from the father, but this allele here, both of them are inherited from the mother. And we know that the chromosome itself stays intact. The chromosome itself is not going to break in half, for example. So our answer to 1.1.6 we know is A. 1.1.7. Which one of the following is correct for speciation through geographic isolation? So we're talking about the arising of a new species, and the thing which has triggered that new species to develop is geographic isolation. So where we have maybe the species developing on 2 different islands separated by the ocean. That's what we call geographic isolation. You may have also learned the term allopatric speciation. That refers to A for away, A for allopatric. The species have developed away from each other. So we're looking for a statement that is correct. The populations undergo phenotypic changes only. And that is very incorrect, because there are no phenotypic changes without genotypic variation as well. Each population undergoes natural selection independently. So on this island, natural selection will be continuing, and on this island, natural selection will be continuing. And because of the ocean, there is no gene flow between the 2 populations, so they are undergoing natural selection independently. I like the answer to B, but let's read C and D just to check. The conditions on each side of the geographic barrier are the same. So the geographic conditions on that island are identical to the geographic conditions on that island. Now, it could be that our two islands do exist under the same geographical conditions, the same environmental conditions, but we know that speciation must take place, and the speciation is going to take place if the conditions are different. All right, so there we've got a problem. The new species formed are genotypically, genetically, the same as the original species. Well, if they're genotypically the same, then it means, quite frankly, they are the same species, because if they were genotypically the same, we could take individuals from one island, reintroduce them to the original population, and they'll be able to carry on having viable offspring. But that is not the case. And so our answer is, as we thought, 1.1.7, B. 1.1.8. Below is the list of events that occur during cell division, and you're given 4 different events. You've got to read through those events and work out which one of the following combinations of events occurs both in meiosis and in mitosis. So option A says you look at 2, 3, and 4, and you decide, yes, that is the combination of characteristics or events that happens in meiosis and mitosis. So the way to answer a question like this is work out from our original conditions or our original list, which are correct, in applying to both meiosis and mitosis. So let's go. Number 1, homologous chromosomes line up at the equator of the cell. That is only typical of meiosis, not mitosis. So number 1 does not apply to mitosis. Chromatids are pulled to opposite poles of the cell. That certainly happens in meiosis and mitosis. Chromosome pairs arrange themselves randomly at the equator of the cell. That happens in meiosis and mitosis. And individual chromosomes line up at the equator of the cell. That's going to happen in meiosis too, as well as mitosis. And so our answer is, in fact, 2, 3, and 4. It's only options with the number 1 in it that are going to be incorrect. And in option D, we're missing number 3, which is correct. So our answer is going to be 1.1.8, A. A short piece of DNA containing 19 nucleotides in each strand. So here we've got strand 1. It must have 19 nucleotides. Strand 2 must have 19 nucleotides. And our strands were analyzed, and the number of some of the different nitrogenous bases is shown. In cytosine, we don't know the number for strand 1, but we know in strand 2 there are 4 cytosine, and so on for guanine, thymine, and adenine. And our question says, how many nucleotides containing thymine, so we're focused on thymine, are present in strand number 1. Now what you need to remember is we don't know on strand number 1 how many thymines are present, but we do know there are 8 adenines present on strand number 1. If there are 8 adenines present on strand number 1, it must mean that there are 8 thymines present because of complementary base pairing. An adenine is always going to join up with a thymine, and a cytosine with complementary base pairing is always going to join up with a guanine, and likewise a guanine with a cytosine. So we're looking at one strand only, and if we have 8 adenines on strand 1, how many thymines will we have? Remember, it's in a 1-to-1 ratio. So we will have 8 thymines. If there are 8 adenines, there will be 8 thymines as well. All right, that's our multiple choice out of the way.